A point mutation is a change to a single nucleotide base in a DNA sequence. Shown here is the unmutated DNA that was given. Below is the mRNA, which matches up exactly with the coding strand, except the thymines are exchanged for uracils. And below that is the normal amino acid sequence. The first example of a point mutation is a missense mutation. Missense mutations are mutations to a single nucleotide in a DNA sequence that cause the amino acid in the translated code to change. For example, we have the original DNA sequence shown here, and below is the mutated one. In the mutated sequence, a cytosine has been changed to an adenine in the third codon. This resulted in the amino acid sequence changing from an asparagine to a lysine at the third codon position. This mutation is an example of a transversion. The original base, cytosine, is a pyrimidine, and the base in the mutated sequence is purine. The next example of a point mutation is a nonsense mutation. Nonsense mutations change a normal amino acid to a stop codon, causing translation to stop prematurely. Shown here is a normal DNA sequence, and below is the mutated DNA sequence. The guanine in the second codon was changed to an adenine. This is an example of a transition because both guanine and adenine are purines. The point mutation changes the amino acid tryptophan to a stop codon. The next example of a point mutation is a silent mutation. Silent mutations change a single nucleotide base in a DNA sequence, but result in a codon that codes for the same amino acid as the unmutated DNA sequence would have. For example, the original DNA sequence is again shown here, as well as the mutated one below it. The cytosine in the fifth codon was changed to a thymine. In the mRNA sequence, this would cause the codon to change from UGC to UGU. Both UGC and UGU code for the amino acid cysteine, so there is no change to the resulting protein. This is an example of a transition because both cytosine and thymine are pyrimidines. Lastly, we have an example of a read-through mutation. Read-through mutations change stop codons into another amino acid, causing the protein to continue to being translated when it would have been stopped before. In this example, the normal DNA sequence has a 6 codon that reads TGA in the coding strand, or UGA in the mRNA strand, which is a stop codon. The mutated DNA sequence has the thymine changed to a cytosine, so the codon is now CGA, which would code for the amino acid arginine instead of a stop codon. The ribosome will continue to create the amino acid chain and read through the sequence to the next codon, even though stop codon belongs in this position. This is an example of a transition because the thymine is changed to a cytosine, and both of these bases are pyrimidines.